Hello, and welcome back to Human Evolution. Today I want to talk about proteins because we've been transcribing DNA and translating mRNA so that we can make proteins, because coding segments of DNA are genes, and genes code for proteins. And just to review, each gene encodes multiple polypeptides, right? So remember, we've moved away from the one gene to one protein because we've got maybe as many as 150,000 proteins, but less than 20,000 genes. And that's where our alternative splicing came in. And then I told you that there were a couple of other things that would allow for proteins to behave differently, right? And so we're going to talk about those other ways today. We've already talked about alternative splicing. One of the big things that we've been really focusing on is that form follows function, right? The idea that something is going to be shaped in a way that facilitates what its purpose is. And so part of the going from just that polypeptide, that chain of amino acids, to a functional protein is how it folds. So the first thing that I want to talk about are the levels of protein structure, and there are 4. First of all, there's the primary structure, and we've really already talked about that. That's just the chain of amino acids. And some proteins, right when they come out, are ready to go, right? And so they only have a primary structure. Lysozyme, which is an enzyme that attacks bacteria, is just a polypeptide chain of about 129 amino acids, and here is a lovely picture of it. But more often than not, right, there's a secondary structure that's really important, and this is going to be governed by the attractions, right, that hydrogen bonding that is going to attract one part of that chain to another part of the chain. And there are 2 big secondary structures. There are the beta pleated sheets and the alpha helices. So a single one of those is an alpha helix. So basically you get these like crinkle kind of situations that are these flat accordion looking things, or you get these spirals of amino acids spiraling around each other. A lot of channels are actually made of this alpha helix structure, so things can go through the center because it makes an opening. But my favorite protein that we want to talk about is spider silk, right? Spider silk is very, very cool, and it is a beta pleated sheet, right? And it is the presence of those bonds, that pleating, that makes spider silk so amazingly strong. Okay, now the next part is about adding stuff to the protein. So you can change the function of a protein by changing its tertiary structure. And the tertiary structure is about the R groups, the functional groups, right? So this is what you're attaching to it, and how those R groups interact with the polypeptide backbone. So you can actually get an amino acid chain to fold in a different way, in a different place, by changing what you've added to it. And this is called protein modification. And here are just some examples of different modifications. Good ones to know are phosphorylation, glycosylation, and ubiquitination. Those are big, and methylation. Those are kind of the big ones that we deal with. We've already talked about when DNA gets methylated, but those are kind of the 4 big ones, and we'll talk a little bit more about those later on. And I'll give you a couple of examples of when they come up. All right, so when does this modification occur? Well, there's kind of 2 arenas. There are the co-translational modifications. These are things that are happening during translation. Folding occurs during this time, and when we watched the video of translation, you could see that the polypeptide that was coming out would start to fold on its own, and that's because of these attractions, right? Sometimes you'll have enzymes that catalyze the protein folding, and then there are a couple other things that happen that we're not going to talk about. But when we talk about this tertiary structure deal, we're talking about adding things, cleaving, right, cutting off pieces, stuff like that. That's all going to be post-translational, and a lot of this actually happens inside the endoplasmic reticulum, because remember, the rough ER is where there are ribosomes on the surface, and then the polypeptide is actually made and goes into the lumen of the endoplasmic reticulum. And we talked about this is where proteins get modified. These are the kinds of modifications that we're talking about. You can cleave off parts. So you remember all of these start with AUG, right? And that was a methionine. You actually may not want that methionine in your final protein, so that might get cut off at this point. And this is where you're going to add all of these other R groups. So, which of the following might not result in a change in the conformation, the form, of a protein? A change in the functional group, a change in the side chain, a change in the amino acid sequence, or a change in the length of the polypeptide? Well, if you remember back to when we were talking about this kind of redundancy thing, just because you change the amino acid sequence doesn't necessarily mean that you're going to change the way something folds, because remember, if the amino acid is in the same chemical group, right, the same chemical family as another amino acid, it'll have a very similar or the same behavior, and so you can actually not have a change to the final protein. There was that, and then there was the idea that multiple codons code for the same amino acid. It was in that conversation that we talked about changing the amino acid sequence can cause a big change, but it might not. Okay, so that was another just little review there. All of these other things, by the way, will change the conformation of a protein. They will change the way that it folds, and therefore they will change the way that it functions. All right, then the last level of protein structure are the quaternary structure, right? This is the quaternary structure. And so what you've got going on here is that you can have more than one protein, right? When you have more than one, they're called polypeptide subunits. You can have more than one joined together to form like a super protein complex. And then this is actually the protein. And a really good example of that is hemoglobin, right? And we'll talk about hemoglobin more when we talk about sickle cell. But hemoglobin is actually 4 proteins that are beta pleated sheets that are then modified, right? And then they come, so that's the tertiary, then they come together and hook together as this unit of 4. So 4 subunits, and then you get this big protein out of these 4 subunits. Another one is collagen, which makes your skin flexible. These are the alpha helix structure, and then you have 3 of them wrap around each other to form a rope, right? And their job is connective tissue. Okay, so just to recap so you can see them all together, there's primary, secondary, tertiary, quaternary. Primary is just how it folds, just coming right out based on the hydrogen bonding. The secondary structure is going to be that bonding between the backbone, right, the polypeptide backbone along regular intervals, giving you the alpha helix or the beta pleated sheet. The tertiary structure is where you modify it with functional groups, and then those interactions change the shape. And the quaternary structure is when you have more than one protein or polypeptide unit forming the protein. And remember, form follows function, right? And in almost every case, the function of protein is going to depend on its ability to recognize and bind to some other molecule. Think about hemoglobin. It's got to bind to oxygen, right? Antibodies have to bind to a foreign substance, and each antibody is unique. That's why it takes a first contact of your immune system with a foreign invader in order to produce those antibodies. Once it recognizes it, it will make specialized antibodies that will attack that particular thing every time it encounters it, right? Enzymes have to recognize a chemical, right, and be able to bind to it so they can facilitate that reaction. So it really matters about how they're formed, because they have to have a special slot that can recognize and bond. Now, on that kind of idea, which of the following is actually not a function of a protein? Building connective tissue, facilitating anabolic and catabolic processes, fighting antigens. An antigen would be something like a bacteria or virus, some sort of foreign invader. Transmitting messages from nerves to cells. All of these are functions of proteins, or none of these are functions of proteins. Well, all of these are functions of proteins. Remember, I always tell you, proteins do everything in your body, okay? So some of the roles of proteins include neurotransmitters. These are transmitting signals from your brain to cells. Enzymes, they're going to facilitate chemical reactions. They either build up stuff or break stuff down. Your whole defense system is all proteins. Your immune system is all proteins. Hormones, regulatory proteins, all of your body processes are triggered and stopped by proteins. Now, they're all your structural proteins, and this is what you think of basically when you think of protein, right? It's like, oh, you got to eat your protein so you can have, like, strong muscles, but it's also the connective tissues. It's also the bones, right? Giving strength to your bones and teeth. It's your hair. It's if you had feathers, you would have those, right? Your fingernails, all of that. And muscle contractions themselves are governed by proteins, actin and myosin in particular. Also transporters. If you want to carry stuff around your body, you're going to need a little protein vehicle to do it. And remember all those signals that were proteins? You've got to have a receptor. If you're going to send a signal, you have to have a receiver. The receivers are proteins. Toxins, all of your toxins. So let's say that you are a venomous snake, right? Your toxins, your venom is a whole collection of lots of different proteins. You can store amino acids for breakdown to make new proteins. You can regulate your fluid balance. The buffers in your blood that keep your blood from becoming too acidic or too basic. And you can actually store energy and glucose if needed. Now that's going to be mostly a carbohydrate thing, but in case of emergency, your body goes straight to proteins and breaks them down. So your daily stuff, energy, is going to be sugar or starch, right? But in emergencies, it'll go to proteins. So all these things. And that is actually it for our first part of our protein conversation. We will be talking about this more in class. And thanks for listening. I will see you next time.